Okay, so Eda, could you introduce our study?
Yeah, of course. We would like to present our study, a new approach to SMA therapeutics with molecular modeling methods.
SMA, or spinal muscular atrophy, is a recessively inherited genetic disease which is caused by the lack of SMM1 gene. The SMM1 gene is responsible of the transcription of the SMM protein, which stands for survival motor neuron. Without the SMM protein, motor neurons cannot survive, and they cannot transfer impulses to muscles, causing them to atrophy, ranging from mild atrophy to severe atrophy. And unfortunately, severe atrophy is mostly observed in infants. And again, unfortunately, SMA is currently the leading genetic cause of death in infants today. Even though there are therapies for SMA currently, they all have the common denominator of being genetically targeted, and this drives up their price, production costs, and research costs. In order to overcome these problems, in our study, we aim to do inhibition simulations of three biological mechanisms, namely ROROC, histone deacetylase, and ubiquitin proteasome pathways using molecular modeling methods in order to unravel potential new drug ligand molecules for the alleviation of SMA symptoms.
So our main method was doing molecular docking with virtual screening methods. Virtual screening is regarded as the selection of likely drug candidates from large libraries of chemical structures by using computational methodologies. And various preparatory steps were performed before starting the VS. You can see like protein and ligand selection, then preparation, finding site definition, and it continues. A total of 6 macromolecules were chosen from our CSP protein databank, and 8,470 ligands were downloaded in the SDF format along with their 3D structures in our study. At the same time, the Zinc-15 work database containing 5,900 ligands approved by various international organizations was obtained in the SDF format for the drug re-evaluation process. The right module of Schrödinger Maestro was used for the docking processes, and where trials were carried out in the high throughput virtual screening HTVS, standard precision SP, and extra precision XP.
After the virtual screening processes and the most feasible ligands were unraveled, we have pharmacologically analyzed those. In the ROC1 protein ligand complexes, it was observed that the isoquinoline rings that you can see right here engaged in hydrogen bonding with the methionine 123 residue of the active site. Likewise, in the MIB1 macromolecule, as you can see right here, the carbazole heterocyclic substructures engaged in hydrogen bonding with the serine 225 residues as well. In the UBA1 E1 kinase molecule, it was observed that there were coincident heterocyclic substructures, and all ligands have engaged in hydrogen bonding with at least one of the arginine 474, aspartic acid 463, and I believe alanine 437 amino acid residues. For the HDAC complexes, it was observed that they all have, without exceptions, engaged in hydrogen bonding with at least one of the threonine residues in the active site. And all ligands in all of the macromolecule complexes were observed to have engaged in strong non-covalent interactions, primarily hydrophobic with the active site residues. After having observed the chemical feasibility of the inhibitions of the macromolecules, we have moved on to the molecular dynamic simulations in our study. After the final in silico confirmations of the proposed ligands inhibitions, we are seeking to move on to real-life confirmations.
And also, among the ligands suggested in our study, those that have therapeutic effects on various neurodegenerative diseases were found. The therapeutic effects on the suggested ligands on different neurodegenerative diseases such as MS, Alzheimer's, Parkinson's, or ALS can be also investigated. So in our study, a total of 6 macromolecules and 14,370 molecules from the derived ligand databases were subjected to virtual screening processes. The final virtual screening trials carried out on Schrödinger Maestro software have yielded 19, 17, and 14 potential drug molecules for the RORAC, HDAC, and ubiquitin proteasome biological mechanisms, respectively. All 50 ligands of interest potential drug molecules have exhibited superior docking score values than the protease native ligands like ligands that are in complex with the protein in their respective PDB entries, and have shown exceptional binding site interactions.
In the virtual screening trials, we have created our own custom ligand databases, and we are also right now in the process of automating the custom database creation in order to increase the effectiveness of our study. We would like to thank Dr. Ersin Günder for helping us learning molecular modeling methods in our study, and our professors from our school and the Schrödinger Incorporated representatives for helping us with all sorts of things and the intricacies of our project, essentially. And here you can see our references, and thank you for listening to us.
Thank you for your attention.
Thank you, Eda and Canberk. This is so impressive that you did as a high school student, whereas many of us still don't know how to do many of these things. If there are any questions from the audience, I can take them up right now. So far nothing. So I'll just ask a very quick question. So you identified some candidate ligands, right?
Yeah.
So how did you verify those ligands that you obtained through the virtual screening methods?
So we actually had 3 steps in the verification process. We first looked at their docking scores that the GLIDE module gives us. Usually, the docking scores for ligands which inhibit the proteins are in the range of -7 to -13. And all of our ligands were actually better than their main ligands, we called them in our study. So the main ligands are essentially the ligands that the proteins were in complex in the PDB entries. And the ligands that we unraveled in our study using our ligand databases actually gave out much better docking results than that. And in the second step, we we looked at their chemical feasibility on 2 dimensions. We looked at their active site interaction diagrams. So like what sort of bonds they engaged in, what sort of interactions they engaged in and with the active site. And for the last step, we looked at their 3D structures, whether they had protein clashes and those sorts of things, how much they have interacted with the solvents in the docking steps. And after that, we classified the ligands as ligands of interest in our study, and we have conducted their pharmacophilical analyses in our study.